ഇങ്ങനെ മാറ്റി വെച്ചാൽ ബാക്കിൽ മറന്നു പോവാ.
ചിലപ്പോ അത് ലോഗോട്ടോ
ചിലപ്പോ മൈക്കോ പോയിരുന്നെന്ന് പറഞ്ഞിട്ട്
അല്ലോ.
Good morning.
Morning, ma'am.
Good morning, ma'am. Only 100 can join.
Yes.
So send your attendance to the chat box. Shall we start?
Yes, ma'am.
So in the last class, we discussed about the glycolysis, its steps, enzymes, the inhibitors, clinical application of this inhibitor, then regulation. One of you summarize. Who will do? Brief summary in 3 or 4 minutes. Ida Hamza, can you summarize? Abdul Karim. When I call out your names, if you're not answering, you will be marked as absent. So Fida is absent. Isa?
Hello, ma'am.
Ah.
We discussed about the glycolytic regulations of glycolysis, metabolism of glucose to pyruvate. Ma'am, so to audible.
Yeah, it's audible.
Yeah, I think it stopped at regulation of glycolysis.
Which are the key regulatory enzymes? Which are the allosteric activators, inhibitors?
In glycolysis, phosphofructokinase, allosteric, ma'am,
Ah,
phosphofructokinase is a allosteric enzyme.
totally, how many regulatory enzymes are there?
Three.
Which are those three?
Phosphofructokinase,
First irreversible step, that is the first key enzyme.
hexokinase,
Exocytosis. Second is the
phosphofructokinase,
and third
pyruvate kinase.
pyruvate kinase. Okay. Which is the inhibitor in the case of hexokinase?
Hexokinase. Ma'am, I don't remember.
Anyone?
Glucose 6-phosphate.
The end product, that is the
Feedback inhibition.
Then, what about the phosphofructokinase? It has got an allosteric regulation. There are allosteric activators and inhibitors.
Inhibitors ATP and citrate.
AMP and?
Activator, allosteric activator.
Okay. Which is the inhibitor?
ATP and citrate inhibit the reaction.
Okay. Which is the activator?
AMP.
AMP. AMP is there. Okay. What about the hormonal regulation? Which hormone activate the pathway of glycolysis?
Insulin.
Insulin. Which is the inhibitory hormone?
Glucagon.
Yeah, it is the glucagon. So I think we have discussed till this way. And one more I discussed about the phosphofructokinase 2. Right. In the glycolytic pathway, the main enzyme is phosphofructokinase 1. Other than that, there is another phosphofructokinase, that is PFK2. When PFK2 is there, there is activation of the glycolysis. Okay. So we discussed about all these steps, the key enzymes, the energy requiring and energy producing steps, and the total energy yield from aerobic cycle and anaerobic cycle. Okay. So what is the total energy yield in anaerobic cycle?
2 ATP.
It's the 2 ATP. What about the aerobic cycle?
7 ATP.
It is 7 ATP. Can you tell when this glycolytic intermediate, that is the pyruvate, if it is entering the TCA cycle, what is the total energy yield?
32 ATP.
It will be 32. How does 32?
7 glycolysis.
And from glycolysis along, then pyruvate dehydrogenase reaction.
5 ATP from.
5 ATP because of 2 NAD. So we'll see that step then. Entering the TCA cycle, how many?
20 ATP.
20. Total of 2 acetyl-CoA will be there. That is entering the TCA cycle from 1 acetyl-CoA, total of 10 ATP, so total 20. Adding all this, we'll get 30. A brief summary of the regulation. You can see the end product, fructose 6-phosphate, can inhibit. Likewise, this step, there is the end products will inhibit. And I told you the glucagon and glucocorticoids, which are the inhibitors of the glycolysis. Likewise, insulin is the activator. When there is plenty of ATP, there is no need for progression of glycolysis. So ATP will inhibit all this key enzyme. Likewise, ADP will activate all this enzyme. Likewise, they have discussed about that is the phosphofructokinase. So when there is active phosphofructokinase, that will also activate the glycolysis by activating phosphofructokinase 2 will in turn activate the phosphofructokinase 1, which is an enzyme in the glycolysis. Okay, the way is like that. The inactive, so you can see the phosphofructokinase 2. Okay, that can activate the phosphatase. Okay, the protein phosphatase, it will in turn activate the phosphofructokinase 1. This picture is not that important. It is there in the Harper. If you want, you can go and read. Otherwise, check the Vasudevan sir textbooks. There is, it is written like this. So I told you, fructose 6-phosphate to fructose 1,6-bisphosphate. This is the glycolytic step. Third step. Here, fructose is converted to fructose 1,6-bisphosphate. The enzyme is PFK1 and it utilizes 180. So, instead of going on directly to the fructose 6-phosphate to fructose 1,6-bisphosphate, it has got another pathway also. Like, instead of adding phosphate to the first carbon, it can add phosphate to second carbon also, forming the fructose 2,6-bisphosphate. Enzyme is phosphofructokinase 2. Okay, when there is excess of glucose, the phosphofructokinase 2 will be activated. And what is the effect of this phosphofructokinase 2 is that will inhibit this enzyme. You'll see this enzyme, the fructose 1,6-bisphosphate. This is a key enzyme in the gluconeogenesis. Okay. So gluconeogenesis will be inhibited by phosphofructokinase 2. Also, it will activate the phosphofructokinase 1, which is a key enzyme of glycolysis. Clear? So, this is the pathway. The reverse arrow is for the gluconeogenesis. This fructose 1,6-bisphosphatase is an enzyme in the gluconeogenesis. You know that the glucokinase or hexokinase, phosphofructokinase and pyruvate kinase, all three enzymes are irreversible. Those steps are irreversible. The enzyme will act only in one direction. Okay. So the phosphofructokinase will convert fructose 6-phosphate to fructose 1,6-bisphosphate only. The reverse will not occur. For the reversal of this thing or for the gluconeogenesis, the enzyme is fructose 1,6-bisphosphatase. Okay. So consider a situation when excess glucose is present in the cell. In that condition, the fructose 6-phosphate, some of the fructose 6-phosphate will go with the help of this enzyme PFK1 to form the fructose 1,6-bisphosphate. Okay. Along with that, there is opening of another pathway which is catalyzed by the enzyme phosphofructose. It is PFK only, but it is another enzyme PFK2. 2 means it is adding phosphate to the second carbon only. Here it is added to the first carbon. Okay. What is the importance of this intermediate fructose 2,6-bisphosphate is that it will inhibit the gluconeogenetic pathway by inhibiting the key enzyme of gluconeogenesis, that is fructose 1,6-bisphosphate. Clear?
Yes, ma'am.
In addition to inhibiting the fructose 1,6-bisphosphate, it also activates PFK1. Okay, so when excess glucose is there, PFK2 will be activated. PFK2 inhibits the gluconeogenic enzyme. Also, it activates the key glycolytic enzyme PFK1. The result is glycolysis will be favored. Okay, that is called reciprocal regulation of the phosphofructokinase 2 by fructose 2,6-bisphosphate by phosphorylation. This is by the phosphorylation of this. Here, silence, please. Others, please mute. Shall I proceed? Please mute yourself. Okay. Shall I move on?
Yes, ma'am.
Okay.
Ma'am.
Yes, ma'am. Okay. Okay, so this is the summary of the regulation of the glycolysis, and you can see the key enzymes are hexokinase or glucokinase. Glucokinase will be present in the which organ? In liver. Whereas hexokinase is present in all cell. Then this is the first enzyme, key enzyme. Second enzyme is the phosphofructokinase. It is the PFK1 only, and pyruvate kinase. And we can say about the pyruvate dehydrogenase also. I'll say about the pyruvate dehydrogenase in the coming slide. I told you the insulin is the one which is activating all the enzymes. Okay, when there is plenty of glucose, the insulin is the hormone which is released to make the glucose enter into the cell and help the glycolysis to proceed. So insulin will, most of this enzyme, so all the 3 glycolytic enzyme will be activated by the insulin, and the glucagon will inhibit all these 3 enzymes. Likewise, I told you the phosphofructokinase, it is an allosteric enzyme, which means there are allosteric activators and allosteric inhibitors. So ATP and citrate, these 2 are the allosteric inhibitors. Okay, and the AMP, an AMP is present, it will be an allosteric activator. Likewise, I discussed about the PFK2. When PFK2 is there by the phosphorylation reaction, that PFK2 will in turn activate the PFK1. So glycolysis will be favored. Okay. So when PFK2 is there, what is formed? It is fructose 2,6-bisphosphate. So we can say another way, when there is increased fructose 2,6-bisphosphate, PFK1 will be favored. Clear?
Yes.
Oh, the same thing about the pyruvate kinase, there will be insulin which is activating the pathway, glucagon. Okay. Likewise, when there is increased energy status or ATP, there will be inhibition of the pathway. Like here, cyclic AMP will also inhibit the pathway. regarding the pyruvate dehydrogenase, we'll discuss in the coming. So about the hormones, I told you insulin is the one which causing the activation of all these three enzymes. How it favors is that it will increase the glucose entry into the cell. When there is increased glucose in the cell, that will favor the glycolysis. Likewise, the reverse anti-insulin hormones are the glucagon and glucocorticoid that will inhibit the pathway. And there is yeah.
Ma'am, YouTube live live chat attendance.
You send the attendance to Parvati. She'll WhatsApp.
Okay.
Okay. So regarding the faster effect, okay, you need to just know what is faster effect when there is Parvati late. എന്താണ് എന്താടോ
അറിയാതെ അൺമ്യൂട്ടഡ് ആയതാന്ന് തോന്നുന്നു.
What is faster effect that it is that when there is plenty of oxygen available, there will be an inhibition of glycolysis, or we can say there is almost a 7-fold decrease in the glycolysis in the presence of excess of oxygen. Okay, just need to know that a faster effect. Okay, what happens when there is plenty of oxygen or there is excess of oxygen is that it will inhibit one of the key enzymes, which is the phosphofructokinase. When I tell phosphofructokinase, it is always PFK1. When it is PFK2, I will specifically tell it is PFK2. Simply PFK means it is PFK1. Okay. And there is another cycle which is occurring in the RBC that is called Rappaport-Luberin cycle, or other ways you can call it 2,3 Vpg shunt. I think you have studied this in plus 2 classes, right? Yes or no? Yes or no?
No, ma'am.
No.
No, ma'am.
Okay, I will tell you what is Vpg shunt. So you know what is the 6th step of glycolysis? Tell me, which is the 6th step of glycolysis? It is an energy producing step, ATP generating step.
1,3 Vpg to 3 Vpg.
Yeah, good. 1,3 Vpg getting converted to 3-phosphoglycerate. There is generation of 1 ATP. Okay, ADP will be converted to ATP. This is the normal 6th step of glycolysis. Instead of this step, to form the 3-PG, 3-phosphoglycerate, there is an alternate pathway. Okay, that is called the Vpg shunt. Is it clear?
Yes, ma'am.
Yes, ma'am. How this pathway happens is that 1,3 Vpg, instead of directly going to 3-PG, it can form 2,3 Vpg. This is a 2,3 Vpg shunt. So instead of 1,3, there is change in the phosphate from the first carbon to the second carbon. So shift in the phosphate group is catalyzed by which enzyme?
Mutase.
Yeah, it is mutase. So 1,3 Vpg mutase is the enzyme which converts 1,3 Vpg to 2,3 Vpg. What happens to 2,3 Vpg is that removal of the phosphate of the second phosphate which is present in the second carbonate. Removal of the phosphate is catalyzed by which hormone?
Kinase.
Okay. Other than that, there is another hormone, phosphatase. Okay, removal of the phosphate. So it is 2,3-BPG. 2,3-bisphosphoglycerate phosphatase will remove the phosphate from the second position so that it will become 3-PG. Actually, 1,3-BPG is converted to 3-PG, but it is occurring in 2 steps, and you can note that this ATP will not be generated if it is going through this Vpg shunt pathway. Is it clear till now?
Yes, ma'am.
Okay. So
Yes, ma'am.
1,3-BPG mutase and 2,3-BPG phosphatase are the 2 enzymes involved in this pathway, and there is no net generation of ATP if glucose is going through this Vpg shunt pathway instead of the normal pathway. Okay. How this removal of phosphate is happening is that there is no ATP involved, so it is not kinase. Instead, there is addition of water and removal of the inorganic phosphate. This is how 2,3-BPG is converted to 3-PG. Clear? So 1,3-bisphosphoglycerate. First and third carbon will be, there will be addition of phosphate. That is 1,3-bisphosphoglycerate. That is converted to 3-phosphoglycerate. That is by phosphoglycerate kinase reaction. 1,3-bisphosphoglycerate kinase reaction, which requires produces 1 ATP. This is the normal pathway, but instead of that, there is a mutase pathway. Instead of the first carbon getting added, phosphate getting added, instead of that, the phosphate is added to the second carbon, forming 2,3-bisphosphoglycerate. It will again get converted to 3-phosphoglycerate by phosphatase. Why it is called phosphatase, not kinase, is that it is no ATP or magnesium is involved in this step, but there is addition of water and removal of the phosphate from this second position. There is no net generation of ATP in this process. This is called a Rappaport-Lubborin cycle or 2,3-BPG shunt pathway. It is a pathway which is usually occurring within the RBC, and normally about 15 to 25% of glucose, instead of going through the normal glycolytic pathway, it may be converted to 2,3-BPG and the cycle moves on. So what will be the effect of this is that there will be no ATP generated. You know that the first step and third step, there will be utilization of 1 ATP. So 1 ATP will be reduced. In the sixth step, what is happening? There will be generation of 2 ATP. Right. This 2,3-BPG, this phosphoglycerate kinase reaction, there is actually generation of 1 ATP, but there are 2 substrates. From 1 glucose, 2 substrates will be there. So 2 ATP will be generated in the 9th step. Pyruvate kinase reaction also, there will be generation of 2 ATP. Okay, right. So this 4 minus 2 is equal to 2. So what happens if it is going to the Vpg shunt pathway is that this 2 ATP will not be generated. So there is 2 minus 2. So no ATP will be generated. That is the drawback of this cycle. No net energy production will be there. Why I am not mentioning about the NADH which is formed in the glycolytic pathway? Can anyone tell?
Electron transport chain.
Why it is not going on to electron transport chain? It is RBC, erythrocyte. Does it have a mitochondria?
No.
So it does not have a mitochondria, and ETC is happening in the mitochondria. So even though NADH is formed, it cannot enter into the ETC to form the 2.5 into 280. So that 5 ATP will not be generated, and you know, if it is going on to 2,3-BPG shunt pathway, this 2 ATP will also not be generated. So 2 minus 2, there is no net generation of ATP. Is it clear?
Yes, ma'am.
Okay. So about the 2,3-BPG, what is the effect of this pathway is that 2,3-BPG will be present in the cell. What happens with 2,3-BPG is that it can combine with the hemoglobin within the RBC. What happens when this 2,3-BPG combine with hemoglobin is that it reduces the affinity towards the oxygen. So oxygen will be removed. HB will be present as HbO2. When 2,3-BPG bounds to it, the 2,3-BPG will get attached to it and oxygen will be released from the hemoglobin. In other words, it helps in unloading of the oxygen. When there is hypoxia, when there is decreased oxygen level in the body, when we go on to the high altitude like that, there is need of oxygen. Oxygen will be decreased so that the hemoglobin should release the oxygen easily from the hemoglobin which is bound to the oxygen should release it readily. So when 2,3-BPG is present in excess, what happens? It will easily remove the oxygen from the hemoglobin. Clear? Yes or no?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. So you will study oxygen dissociation curve, ODC, in physiology under respiration. What happens with this is the normal sigmoid shape oxygen dissociation curve. This is the partial pressure of oxygen, and this is the release of the oxygen. Okay, so when there is 2,3-BPG, what is the effect of 2,3-BPG on the oxygen dissociation curve is that it will shift the curve towards the right. This is the original curve. When there is increased 2,3-BPG, the curve will be shifted to the right, which means that there will be easily dissociation of oxygen from the hemoglobin. Okay, deoxygenation will be promoted, and that will be beneficial in high altitudes. Okay, in which all conditions it will increase is that in hypoxic condition, one is high altitude, second is fetal circulation. Both this condition you must know, high altitude and fetal circulation. In both this condition, 2,3-BPG level will increase. What is the effect is that it will bind to the hemoglobin and help in the release of the oxygen from the hemoglobin. Okay, this is about the 2,3-BPG shunt pathway. And regarding the clinical effects like enzyme deficiency. Actually, the enzyme deficiency of the key enzymes or other enzyme in the glycolytic pathway are rare. These are rare disorders. Deficiency commonly leads to depletion of RBC. Why? Depletion of specifically RBCs.
Only energy production in RBCs through.
RBCs depend solely on the glycolysis as it does not have mitochondria for the energy production. Okay, but so when there is enzyme deficiency, RBCs are devoid of energy or ATP. So lifespan of RBC will be reduced. There will be increased destruction of RBC. So there will be depletion of RBC. So what is the effect of depletion of RBCs is that RBC normal lifespan is 120 days, but if it is devoid of energy or ATP, the lifespan will be, they will be destroyed faster. So there will be increased breakdown of RBC causing hemolytic anemia. Hemolytic anemia. Okay. And hexokinase deficiency. When there is hexokinase deficiency, there is 2,3 BPG level in the RBCs will be decreased. So when the what is the effect of increased 2,3 BPG? That will allow deoxygenation or supply of oxygen to the RBC. When there is decreased 2,3 BPG, what happens? The deoxygenation won't be favored. The oxygen will not be delivered to RBC. Clear? There will be high affinity of oxygen. Likewise, when there is pyruvate kinase deficiency, there will be high 2,3 BPG level in the RBC. High 2,3 BPG level in the RBC means always hemoglobin, there is dissociation of oxygen. Okay. It cannot carry oxygen for long distance because it will be dissociated from the hemoglobin. Pyruvate kinase deficiency, there will be high 2,3 BPG. In hexokinase deficiency, there will be low 2,3 BPG in the RBCs. So low affinity for oxygen. This kind of hemoglobin is not suitable for transport of oxygen to the tissues. Next one is the aldolase deficiency. Here also there will be hemolytic anemia. What happens in phosphofructokinase deficiency is that there will be decreased exercise capacity. Okay. For the exercise to happen, the muscles need energy. When there is energy depletion, anaerobic pathway will be activated and lactate will be formed. Okay, when there is this enzyme deficiency, the exercise capacity will be phosphofructokinase deficiency. There will be decreased exercise capacity, and muscle weakness will be there. And these symptoms will be more pronounced on high carbohydrate diet. When there is high carbohydrate diet only, we need this glycolytic enzyme. So the symptoms will be highly pronounced on high carbohydrate diet, and that will be improved with the lipid diet. Okay. Then moving on to the next thing, which is the pyruvate. In the glycolytic reaction, we have seen about the 9th step. The last one which is formed is the pyruvate. If it is an anaerobic reaction, glycolysis, it is the lactate that will be produced. Let's see what are the fates of pyruvate. Okay, you can see the glucose will be converted to phosphoenol pyruvate in glycolysis. And the last step of glycolysis, it is a pyruvate kinase reaction forming the pyruvate. Okay, pyruvate can be called as a junction point. Why? Because from the pyruvate, different intermediates can be generated. This thing you already know. It is a lactate dehydrogenase reaction. When there is a anaerobic cycle or the oxygen availability is less, the pyruvate will be converted to lactate, utilizing what? Utilizing what?
NADH.
NADH formed in the glycolytic initial step, that will be utilized to produce the lactate with the help of the lactate dehydrogenase enzyme. So this is the first fate of the pyruvate. Or it can be transaminated to alanine. Okay, transaminated to alanine, which means the transfer of amino group. Okay, transfer of amino group. We'll study this under the amino acid metabolism and chemistry. Was it taken by Dr. Jusha? Transamination reaction. Please respond. Alanine to pyruvate reaction. So alanine is an amino acid. Okay. Alanine can combine with a keto acid. Example of keto acid is alpha ketoglutarate. We'll study the details in the amino acid metabolism. An amino acid can combine with the keto acid to form another keto acid and another amino acid. Another amino acid is glutamate. Okay. So this alpha ketoglutarate, it has got a keto group that will accept the amino group. Alanine has got an amino group. That amino group is accepted by the alpha ketoglutarate to form the glutamate, and the rest of the thing remaining is the pyruvate. Is it clear?
Yes, ma'am.
Yes, sir.
That kind of reactions is called transamination reaction. It is the alanine getting transaminated, so it is called alanine aminotransferase. ALT means AL is alanine, alanine aminotransferase. Okay, what is the need of this reaction here in carbohydrate metabolism is that when the glucose supply is in short, body needs glucose continuously because RBC and brain needs a continuous supply of carbohydrate glucose. So from the amino acid, glucogenic amino acid like alanine, gluconeogenesis can be turned on. So alanine can be a substrate for gluconeogenesis. Alanine can be first converted to pyruvate, and finally partial reversal of glycolysis happen to form the glucose. Got a vague idea?
Yes,
Yes
ma'am.
or no? Shall I
Yes,
move on?
ma'am. Yes, ma'am.
Okay, then next is the pathway which we'll be discussing in the coming slide. This is a pyruvate dehydrogenase reaction. I told you 2 NADH will be from one pyruvate, 1 NADH. 2 pyruvate will be formed from 1 glucose, so 2 NADH will be formed. A total of 5 ATP will be formed. Pyruvate to acetyl-CoA reaction, and this acetyl-CoA can enter into the citric acid cycle to form the ATP. So this is the second phase. Third phase. First phase is the anaerobic reaction. Second phase, I told you, transamination reaction. Third phase, I told you, the pyruvate dehydrogenase reaction to finally enter the TCA cycle or the citric acid cycle. Okay. In addition to that, there are other cycles also. Okay. So okay. Then pyruvate, instead of going on to these three things, it has a fourth way, that is the pyruvate carboxylase reaction, carboxylation reaction. What do you mean by carboxylation is that addition of carbon dioxide, it needs a coenzyme, that is biotin. That is a B complex vitamin, biotin. Biotin dependent carboxylation to form the oxaloacetate. So reaction is pyruvate carboxylase reaction or the carboxylation reaction. What is the need for this formation of oxaloacetate is that I told you there are 3 key enzymes for glycolysis. One of the key enzyme is pyruvate kinase reaction, which means that this reaction will occur in one direction only. So for the gluconeogenesis to function, this pyruvate is formed. It cannot be reversed to phosphoenol pyruvate directly by the reversal. For that, we need this step should be circumvented by another set of enzyme. One enzyme, we cannot directly convert pyruvate to phosphoenol pyruvate in a single step. For that, we need 2-step reaction. For that, the first step is the pyruvate carboxylase reaction. Pyruvate is a 3-carbon compound. To that, 1 more carbon is added from the carbon dioxide to form the oxaloacetate, which is a 4-carbon compound. And that reaction occurs within the mitochondria. I told you up to the pyruvate, the reactions are happening in which organelle?
Cytoplasm.
It is in the cytoplasm. But the pyruvate to oxaloacetate conversion is occurring in the mitochondria. Pyruvate can easily enter through the membrane of the mitochondria to reach inside of the mitochondria, and the enzyme is located within the mitochondria. It can convert the pyruvate to oxaloacetate. Clear?
Yes, ma'am.
Okay. And the oxaloacetate can form the rest of the, only this reaction is occurring in the mitochondria. The rest of the reactions are happening within the cytoplasm. The oxaloacetate can be converted to malate with the help of an enzyme, malate dehydrogenase. Details you'll study later. And what is another fate of malate is that malate is also an intermediate in the TCA cycle. It can enter into the cycle. Okay. And the another effect of oxaloacetate is that it is also a transamination can be possible. Amino group can be transferred from the aspartate to form aspartate plus alpha ketoglutarate to form oxaloacetate and glutamate. Actually, it is aspartate that is getting AS aspartate. Aspartate is getting transaminated, so it is the enzyme is AST. Okay, aspartate. These reactions are reversible reaction. That is why I am telling in the other order, aspartate to oxaloacetate. Okay, so the another fate of oxaloacetate is either it can be converted to malate or it can be converted to aspartate. Okay. So the main purpose of this pyruvate carboxylase reaction is forming oxaloacetate, is I told you, to circumvent this pyruvate kinase reaction directly. There is no enzyme which convert the pyruvate to phosphoenol pyruvate. So it is occurred through two steps. One is pyruvate to oxaloacetate with the help of pyruvate carboxylase reaction. The second is the oxaloacetate that is converted to phosphoenol pyruvate, that is by the enzyme phosphoenol pyruvate carboxykinase. Okay, so instead of pyruvate kinase in the glycolysis, to circumvent that, there are two enzymes. One is pyruvate carboxylase reaction, which is occurring in the mitochondria, and the second one is the phosphoenol pyruvate carboxykinase, which convert the pyruvate oxaloacetate to phosphoenol pyruvate. Is it clear till now? Okay. This is the summary of the fates of pyruvate. Pyruvate can be called as a junction point because several reactions are possible from the pyruvate depending upon the energy status. If it is an anaerobic pathway, lactate will be formed. If the glucose supply isn't short, from the glucogenic amino acid, pyruvate can be generated. That is by the transamination reaction. And the pyruvate, the energy status, if glucose is available in primary, glycolysis will be favored and the glucose can be channeled to the TCA cycle through pyruvate dehydrogenase reaction, or it can be channeled to the gluconeogenetic pathway. To circumvent the pyruvate kinase reaction, there are two enzymes, pyruvate carboxylase and phosphoenol pyruvate carboxykinase, to convert to phosphoenol pyruvate. Okay, shall I move on? മനസ്സിലാവുന്നില്ലേ?
Yes, ma'am.
Yes, ma'am.
Okay. This is the same thing we have discussed just now. So in anaerobic condition, what will be favored? Pyruvate to lactate will be favored, and there is utilization of 1 NADH. Okay, this is the first fate. So pyruvate will be converted to lactate in actively contracting muscles. There is continuous need of this energy. So pyruvate, anaerobic pathway will be activated to form the lactate. Excess of lactic acid when accumulated in the muscle, it can result in muscle cramps. That is the reason for muscle cramps when there is a when the muscles are actively contracted. And the lactic acid formed in the body or the muscle can be removed by the Cori cycle. I will tell you what is Cori cycle. So what happens in the muscles is that glucose is converted to pyruvate. But if the muscle is contracting for a long period, which means that all the oxygen supply will get depleted. So muscle have to depend upon the anaerobic glycolysis, which means the pyruvate will be converted to lactate, and there will be increased accumulation of lactate, which is the reason for
Fatigue.
It is the reason for muscle cramps and fatigue. And the lactate formed cannot be converted to glucose directly within the muscle because the gluconeogenetic pathway is occurring in the liver only. So what can happen is that the lactate can enter into the liver via blood, and the lactate which is reaching the liver can get converted to pyruvate, and this pyruvate can be converted by glucose by gluconeogenesis. So the accumulation of lactate can be decreased to a certain amount by this Cori cycle. Cori cycle means that the lactate which is produced in the muscle, preventing the excess accumulation of lactate is by transport of the lactate via blood into the liver, and in the liver it is converted to glucose by gluconeogenetic enzymes. Finally, this glucose can be reutilized by the muscle for the energy. Clear?
Yes, ma'am.
Okay. So it is an energy needing procedure, and energy provided is by the increased metabolism. Aerobic condition, pyruvate is converted to acetyl coenzyme. Pyruvate synthesis up to that is occurring in the cytoplasm, and there is a pyruvate transporter. I told you pyruvate can easily enter into the mitochondria. That is with the help of a transporter which is present in the mitochondrial membrane. And the main fate, I told you, pyruvate is decarboxylated. There is oxidative decarboxylation. That is the term you should know. It is oxidatively decarboxylated by the enzyme pyruvate dehydrogenase. Okay, coming to the pyruvate dehydrogenase complex. Pyruvate dehydrogenase, it is a multi-enzyme complex. You study under enzymology what is multi-enzyme complex, which means that the single enzyme pyruvate dehydrogenase complex has got 3 separate enzymes within it. One enzyme complex has got 3 enzymes and 5 coenzymes. The assembly of 3 enzymes and 5 coenzymes here is called the multi-enzyme complex of the pyruvate dehydrogenase. Let's see which are these 3 enzymes. One is pyruvate dehydrogenase itself. Second is the dihydrolipoyl transacetylase. And the third is dihydrolipoyl dehydrogenase. These are the 3 enzymes which are present in the multi-enzyme complex of pyruvate dehydrogenase. This thing is very important for you. Pyruvate dehydrogenase can itself be asked, so you have to tell that it is a multi-enzyme complex. It consists of 3 individual enzymes and 5 coenzymes. I'll show you what are the 5 coenzyme. One is the thymine pyrophosphate. We can say it as TPP. Second is the thymine is the, what is thymine?
Vitamin
Vitamin B3.
vitamin
Vitamin B1 is the thymine. Then lipomide is the second coenzyme. Third one is the coenzyme A. Then next is the FAD, which is derived from the vitamin B complex riboflavin. And there is another coenzyme, niacin, which is derived from the vitamin B complex niacin. It is the NAD. So these 5 coenzymes are essential for the action of 3 enzymes. Which are those 3 individual enzymes? One is pyruvate dehydrogenase itself. The second is dihydrolipoyl
transacetylase.
Next is the dihydrolipoyl
Dehydrogenase.
Okay. So 3 enzymes and 3 coenzymes should be non-violent. Okay. I will describe the steps shortly. So this is the pyruvate. I told you it is a 3-carbon compound with a keto group. Okay. First step is pyruvate dehydrogenase. So it is, I told you what is the reaction happening. There is oxidative decarboxylation. So first step, there is decarboxylation, removal of the carbon dioxide. So this COO, carboxylic acid group, is removed as carbon dioxide. And this, for the pyruvate dehydrogenase enzyme, the coenzyme is the thymine pyrophosphate. To the thymine pyrophosphate, hydrogen will be added. Okay, this carbon dioxide will be removed as COO minus. The remaining is CH3C double bond O. To that, one hydrogen is added on these 2 sides to remove this double bond. So the product formed is hydroxy. 2 carbon is there. Hydroxyethyl thymine pyrophosphate. This is the first reaction in the multi-enzyme complex. Is it clear?
Yes, ma'am.
Okay. Coming to the second enzyme, that is the dihydrolipoyl transacetylase. Here what is needed is lipomide is a second coenzyme. Here we need an oxidized lipomide. The structure of oxidized lipomide, L means lipomide. It has got a disulfide bond. Okay. So oxidized lipomide can react to this product of the pyruvate dehydrogenase reaction, and this oxidized lipomide can be reduced, which means this disulfide bond can be broken down and form CH3CO. The CH3CO can be added to this lipomide, oxidized lipomide, forming, this is a reduced lipomide. Okay, this can be a CH3CO can be added to this SL and SH. This H is also there. Okay, so it forms acetyl lipomide. 2 carbon means it is acetyl. Acetyl lipomide is formed. This is the second reaction which is catalyzed by the second enzyme of the multi-enzyme complex, dihydrolipoyl transacetyl. Is it clear till now? Actually, these structures are not important for you, but for the better understanding, I'm showing with the help of a structure. Otherwise, you need to know only first there is a carboxylation, removal of carbon dioxide. Carboxylation is there, TPP is involved forming hydroxyethyl TPP. Then second is oxidized lipomide is added to that, forming a reduced lipomide, which is acetyl lipomide. To that, coenzyme A. The third coenzyme I told you is the coenzyme A. Coenzyme is written with SH, means SH is the reactive group. Sulfhydryl group is the reactive group. So it is written separately, coenzyme A. It means there is an SH group. So coenzyme A also react to that to form the acetyl coenzyme. This is the product which we need. for entering into the TCA cycle. Okay. So this is the dihydrolipoyl transacetylase reaction. As a continuation of this, there is addition of coenzyme A to form acetylcoenzyme A and dihydrolipomide. So you can see this is a reduced lipomide. To become oxidized, there should be a double bond. Okay. So how will it happen is that the third enzyme of the multi-enzyme complex will act, dihydrolipoyl dehydrogenase. Okay, that will accept these two hydrogen. Coenzyme is the FAD. FAD will accept these two hydrogen to form the FADH2. So this oxidized lipomide will be regenerated. And what happens to FAD is that we need a continuous supply of FAD so that the reduced lipomide should get converted to oxidized lipomide. So what happens, this FADH2 once formed, it should also get reconverted back to FAD, which means that there should be an H2 acceptor. That will be the next fifth coenzyme, that is the NAD. So this NAD will accept this H2 hydrogen to form the NADH. What is the fate of NADH? It will go on to TCA cycle, finally enter the ETC to form 2.58. Is it clear till now?
Yes, ma'am.
Yes or no?
Madam, can you explain once more?
Okay. From the very first step.
Yes, madam.
Okay. So this is the pyruvate. It is a 3-carbon compound which has got a keto group. It will combine with TPP, which is the first coenzyme. Okay. So TPP will accept this thing only. And this part will be removed as carbon dioxide. That is called oxidative decarboxylation. Okay. It will form hydroxyethyl. 2-carbon alcohol is called ethanol. Right. So there is a hydroxyethyl TPP is there. You have class at 9?
Yes, ma'am.
Which class?
Physiology.
Physiology. Okay. Let me take 5 more minutes. I'll finish now. Okay. So hydroxyethyl TPP will be formed. This hydroxyethyl TPP with the help of second coenzyme. This is the first coenzyme TPP. Second is the oxidized lipomide. Oxidized lipomide will combine with this hydroxyethyl TPP to form acetyl lipomide. Okay, 2-carbon compound we can, if it is an acid, which we can call it as acetic acid. So CH3COH will be acetic acid. So we can say it as acetyl group. So acetyl group combined with this lipomide to form acetyl lipomide. Okay. Next step is, which is catalyzed by dihydrolipoyl transacetylase, which is the second enzyme in the multi-enzyme complex. Third step is the addition of coenzyme A. Actually, what we need is from pyruvate to acetyl-CoA. That acetyl-CoA is formed with the help of the second enzyme and coenzyme A to form the acetyl-coenzyme. Why the rest of the reactions are happening is that we need a continuous supply of the oxidized lipomide. Then only this hydroxyethyl TPP will be converted to acetyl lipomide. Clear?
Yes, ma'am. Yes.
Okay. So here you can see the one of the other products after acetyl coenzyme A is the dihydrolipomide, which is a reduced lipomide. Oxidized lipomide means there should be a double bond. There should be a bond between 2 sulfur groups. Disulfide bond. Okay. So here there is no such bond. To create that bond, we have to remove this hydrogen. That is removed by dihydrolipoyl dehydrogenase. Removal of hydrogen by dehydrogenase. Then we can create a bond between two sulfur groups. That removed hydrogen is accepted by the next coenzyme. The fourth coenzyme is FAD. FAD will accept it to form FADH2. What is the problem is the FADH2 is that it cannot remain in that state because there should be a continuous supply of FAD so that the dihydrolipomate or the reduced lipomate should be converted to oxidized lipomate. So the formed FADH2 will be again reconverted to FAD, which means that the hydrogen should be removed from the FADH2. The removed hydrogen will be accepted by the next coenzyme with the fifth coenzyme, that is NAD, to form the NADH. What is the fate of the NAD is that NAD will enter into the TCA cycle, ETC to get the TCA cycle. It can directly enter into the ETC to form the AT. Is that clear?
Yes, ma'am. Thank you.
Okay. So rest of the thing we'll discuss in the next class tomorrow at 9 o'clock. So read thoroughly and come. I will ask question. If you're not answering, I will mark you absent. Any doubts also you can ask tomorrow. Thank you.
Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Hello, am I audible?
Yes, ma'am.
Please check if somebody's mic is left on. There is some disturbance. Now, in case there is any problem with the either volume or the video visuals, representative can give me a call. Is the screen visible to all?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am. I see there are only 72 participants or 73. Why is it that?
Rest are watching on YouTube, ma'am. Live stream.
But actually 100 can watch live. Then why only when it is 100 then you need to go to the link, isn't it?
Yes, ma'am.
Okay, whatever. So we are continuing with Neuron. Today your first specific learning objectives are to know the properties of a neuron. Now, basically, what property of neuron can you mention? Any property that you can mention? What is basically a neuron? How does it respond?
Excitability.
Excitability, yes, exactly. That is a major property. A nerve responds to a stimulus and is excited. This excitability is the first property that it responds to a stimuli. Now, in relation to the excitability, we will discuss a strength duration curve just after we list out the properties. The second also something that we have discussed previously. is all or none law. Now, can you just recollect? Can one of you recollect what is this all or none law? Now, one of your mic is on. Can you just yes, now the disturbance is okay, clear. So all or none law, it basically refers to a property of stimuli, what we call threshold stimuli. So the minimum intensity of a stimulus which produces a response is threshold stimuli. So if a threshold stimulus is applied, all the stimuli are going to produce the action potential. So the response is produced. If it is below the threshold, there is no response at all. This is what we had discussed in last class. The third property is refractory period. Now, do you remember the assignment or the imposition that you had to submit regarding refractory period? It was to be submitted on Wednesday. So can you say what is refractory period? Yes, what is refractory period?
After the application of first stimulus, duration for which the neuron is unresponsive to the second stimulus.
Okay. So when a second stimulus is applied, there is a duration during which the nerve is not responsive. You are able to see the screen, isn't it?
Yes, ma'am.
Okay. The fourth property is accommodation that we shall now discuss, where a slowly increasing strength of stimuli fails to produce a response, what we call reduced excitability of the neuron. The fifth property is infatigability. Now, it is quite surprising to know that a nerve does not undergo fatigue. Now, do you believe that a nerve does not undergo fatigue? You keep on reading, you are using a lot of your neuronal activity, but still you feel tired. So where is the fatigue occurring? Your neurons do not undergo fatigue. That aspect also we shall discuss today. Sixth property of a nerve, it is conductivity, that is ability to propagate a change in the membrane potential, which is the action potential. And it is continuously regenerated. The is adding up effect or summation. This is seen at the level of dendrites and soma. Piece that we have just mentioned here, which is accommodation. Now coming to what is accommodation, when you apply a slowly rising strength of stimuli, here we refer to an electrical stimulus to a nerve. Generally, the nerve adapts and there is no action potential generated. A response is not seen. Why is it that a slowly rising strength of stimuli is not producing an action potential? Now, basically, which ion change do you require for producing action potential? Which is the ion change responsible for depolarization?
Sodium.
Sodium influx.
Lots of
sodium.
Exactly. So it is the voltage gated sodium influx that is required. Now, when a slowly rising strength of stimulus is applied, there is slow opening up of the voltage gated sodium channels. Now, slowly sodium is getting in, but then there is another change occurring along with it. There is potassium efflux also occurring via the voltage gated potassium channels. So the entry of positive ion into the neuron is counterbalanced by the positive ion potassium moving out of the cell. So the interior depolarization does not occur. Therefore, action potential is not generated. Is that aspect clear regarding what is accommodation?
Yes, ma'am.
Okay. So basically, a nerve is adapting and there is no response seen. Now coming to the question, why is that the neurons do not undergo fatigue? Now what do you mean by fatigue? When you are repeatedly going on doing some activity, say you are running a lot, you feel very tired, fatigued. Thereafter, you are not able to do that activity. That is simply explaining what you mean by fatigue. Now, why is that the neurons can repeatedly transmit the action potential, but still they will not undergo fatigue? The answer lies in refractory period itself. This is a duration during which a second stimuli does not produce a response in the neuron. Also, whenever we say an action potential is being generated and conducted, it is not the same action potential each time. It is a new action potential, new ionic flux or sodium influx that sets up the new action potential. Therefore, neurons will not undergo fatigue. Then why is it that as you keep reading a lot, you're doing a lot of neuronal activity, still you feel quite tired? Well, the site of fatigue in your nervous system, it is the synapse, which is the junction between the two neurons. And here, it is the exhaustion of neurotransmitter, the chemical neurotransmitter that leads to the fatigue, reduced activity. Now, one is also mentioned for the fatigue at the synapse, that is the inactivation of the receptors which are present in the postsynaptic neuronal membrane. So basic reason of neurons not undergoing fatigue is that it is having a refractory period. At least now I hope you realize why this is given as an assignment for you to submit on Wednesday. Refractory period is very important. Now, what does this diagram show? If the RMP is shown as minus 70 millivolts, you are having a diagram in which there is a time scale and the amplitude on y-axis shows millivolt. What is this diagram?
Action potential.
Action potential.
So this is the action potential of a neuron or a nerve. Now, if you are asked, what is the extent of the absolute refractory period? It is extending from the point here, firing level, up to 1/3 of the repolarization. And from there, till the start of the after depolarization is the relative refractory period. Now, the reason for the refractory period lies in the voltage gated sodium channels. So, which is the gate which remains closed and is responsible for refractory period? Which gate of sodium channel?
Inactivation.
Inactivation gate, is it the outer gate or the inner gate?
Inner gate.
Yes, actually with the name itself you can remember it, inactivation. So it is the inner gate. That way you wouldn't go wrong. Now moving to one of the properties that we had listed out along with the neuron. This is called a strength duration curve. Now this is something which has been repeatedly asked for your exams to draw and explain what do you mean by the strength duration curve. This is a curve which shows the relation between the strength of a stimulus and the duration of a stimulus. And what we are referring to is a threshold stimulus. Keep in mind that minimal intensity of stimulus which produces a response, that is threshold stimulus. So we are going to discuss graph which will have two axes, one showing the strength of threshold stimulus, second showing duration. In that, the first term that you need to know, you may be new to this term called Rio base. The term Rio base, it refers to the minimal strength of stimulus which is required to produce an action potential. This is in the strength duration curve. That you are seeing on the x-axis, you can see the time scale. The duration is mentioned in milliseconds. On the y-axis, the stimulus strength is mentioned in millivolts. Now you are seeing a curved line, and the y-axis shows strength of stimulus. So we just mentioned rheobase as the minimum strength of stimulus which is required to produce a response. Now we are just tracing a dotted line to meet this curve here. So on the y-axis, that minimum strength or this amplitude of voltage which is required to produce a response, this is called rheobase. Is the term clearer now?
Yes.
Now on the x-axis, we have the time scale. Now that minimal stimulus or rheobase, for what duration or for what time it is to be applied to produce a response, this is called utilization time. So that minimal strength is rheobase. The time for which you have to apply it, that is the utilization time. Are the two aspects clearer now?
Yes, ma'am.
So utilization time, it is a duration for which a rheobase strength stimulus is applied to produce action potential. Now, since the names are different, there is no other way than memorizing it. But then you have to understand the basic concept here. It is a strength of stimuli, that minimal strength that we call rheobase, and the time for which it has to be applied to produce action potential, that is called utilization time. So those two, we are now clear in the graph. Now, if this is the strength of the rheobase stimuli, suppose we consider double the strength of the rheobase stimuli, when will it produce a response? That's the next aspect we are going to see. Double the rheobase strength stimuli, for what duration it has to be applied to produce a response, that is called chronaxy. Now, this is also a new term. Chronaxy, it refers to the time duration for which a double the rheobase strength stimulus has to be applied to produce an action potential. So chronaxy is the time. So just look at the diagram again. On the time scale, we are seeing for how much duration, what strength of stimuli, double the rheobase strength stimuli has to be applied. That duration is called chronaxy. Are the three terms clear now?
Yes, ma'am.
Is rheobase is the minimal strength of stimuli to produce a response. The duration for which a rheobase strength stimulus is applied is the utilization time. Duration for which a double the rheobase strength stimuli has to be applied is chronaxy. Naturally, if the stimulus strength is doubled, it has to be applied for a shorter duration of time only to produce a response. This is the interpretation of this curve that we see. And this is the strength duration curve. So if it is asked as a question, you have to draw the graph and you have to explain what is rheobase, what is utilization time, and what is chronaxy. Now chronaxy, since it is a duration for which the double rheobase strength is to be applied, it actually is more important. Why? Because it gives you an idea of the excitability of a tissue. Suppose the chronaxy is lesser. For lesser duration, you have to apply the double rheobase strength stimulus. The excitability of the tissue is greater. So lesser the chronaxy, greater is the excitability. That is one information you can get from the strength duration curve. I know these terms are newer and little bit difficult, but I hope at least the graph you will be able to explain. If that much is clear, we shall move on.
Yes, ma'am.
So your next specific learning objective is to know the structure, the transmission of and the blockers in neuromuscular junction. Just a minute, just a minute. Okay, sorry for the disturbance. You are able to see the screen?
Yes, ma'am.
Okay. Now, you need to know the structure, the transmission across neuromuscular junction, and few blockers of transmission across the junction. Now, neuromuscular junction, from the name itself, you know, it is a junction of a nerve on the muscle. It's also known simply as a myoneural junction. It's a junction between a motor neuron and a muscle fiber, which produces the contraction of muscle fiber and leads on to activities or movements. Now, the neuromuscular junction, which is a synapse between a nerve and a muscle, is a chemical synapse. So it is a chemical neurotransmitter which is going to produce the effects. And there is a gap between the neuron and the muscle. The neuronal membrane is separated from the muscle membrane called sarcolemma. And that gap, as you know, is synaptic cleft. So if you are asked, what are the three basic parts of a synapse? There is a presynaptic part, a synaptic cleft, which is a small gap between the two, and a postsynaptic part. This is a diagram in which you can see a presynaptic neuron. Its terminal ending is enlarged to form a bulb-like structure, which is called the presynaptic nerve or the terminal button. This is an enlarged part. And it ends on the next postsynaptic cell. Now, if it is a neuron, it is a synapse between two neurons. If it is a muscle, it is a neuromuscular junction. So you have a presynaptic neuron, a synaptic cleft, and the postsynaptic cell. Now, if the postsynaptic cell actually is a neuron, there are some dense areas on which receptors will be present. It's called the postsynaptic density. The gap between the two presynaptic and the postsynaptic cell is called the synaptic cleft. Transmission occur. Action potential, it comes along the axon of a neuron, reaches the terminal endings of a neuron. So, and voltage change is what is transmitted down the membrane. Now, in the presynaptic neuronal membrane, there are some areas called active zones where synaptic vesicles are present in more numbers. What synaptic vesicles contain actually?
Neurotransmitters.
They contain the chemical neurotransmitter. These are membrane-lined spherical structures. What do you mean by synaptic vesicles? Now, potential actually reaches the membrane terminal. It's a voltage change in the membrane which is propagated down. This leads to opening up of voltage-gated calcium channels. Calcium influx occurs into the neuron, and this calcium is required for the synaptic vesicle to exocytose its contents into the synaptic cleft. So with the help of calcium, the synaptic vesicle fuses with the presynaptic membrane, which undergoes exocytosis. Now somebody's mic is on. Please ensure your mic is muted, except for asking any doubt. Please check somebody's mic is on. It creates disturbance. Okay, is the screen visible now?
Yes, ma'am.
Okay. Now, once there is exocytosis of the neurotransmitter, which is acetylcholine, it then diffuses and binds to receptors which are present on the postsynaptic membrane. And the receptors are nicotinic cholinergic receptors. Now, this is generally what is happening in the neuromuscular junction. Now, there are a few more details that we are going to look into. This is just the basic idea of what is happening. Now, we are going to discuss the neuromuscular junction. So, the first component that we are discussing is a presynaptic neuron. Now, you know, this is the terminal ending of a motor neuron, and the axon terminal ending, when it is terminating, making a synapse with the muscle part, this ending expands, and the expanded portion is nerve-like, so it is called a synaptic nerve or terminal button. And usually this is sitting in a small groove of the muscle membrane, and that groove is called synaptic trough. So it's just sitting in a depression, but there is a gap between the two, which is the synaptic cleft. Now, something to be noted here is that there is plenty of mitochondria in the presynaptic ending, in the synaptic nerve. So why do you think we need mitochondria? A lot of active processes are going to occur here. So you require ATP. So that itself is indicated by the presence of plenty mitochondria. And in the synaptic nerve, you are having synaptic vesicles. They are filled with the neurotransmitter acetylcholine. Now this neurotransmitter, it is synthesized in the presynaptic neuron's cell body, and then it is transported down by fast axonal transport and reaches the nerve terminal. Just a point to know that there is a glial cell which covers the terminal part. It's a Schwann cell and it is called teloglia. Now, if you are reading 3 or 4 textbooks, you will notice that in some books there is mention of this term teloglia. It's just a type of Schwann cell. So the most important feature that you have to mention in the presynaptic neuron, that its terminal ending is modified to form the synaptic nerve or terminal button. It contains these spherical structures which contain neurotransmitter called synaptic vesicles. Now this synaptic vesicle, where is it actually coming and sitting on the membrane of the presynaptic neuron ending? It usually comes and stays docked, just like ships are going to dock. Like that, the vesicles come and they attach to some areas on the membrane of the neuron. These areas are called active zones. In these active zones, there are some modifications occurring which form dense bars, and it is these dense bars that contain voltage gated calcium channels. It is enough to know that synaptic vesicles come and they dock at the active sites, and these active sites, they are having voltage gated calcium channels. So when an action potential which is transmitted down the neuron, presynaptic neuron. It reaches a terminal ending membrane. The voltage change is occurring. This leads to opening up of voltage gated calcium channels. So calcium enters. This calcium influx is required for exocytosis. Now, what is basically happening when we say this exocytosis? Now, in this diagram, here is a synaptic vesicle. This synaptic vesicle comes and docks at active sites. Thereafter, when calcium, with the help of this calcium, the synaptic vesicle has fused with the neuronal membrane, and by exocytosis, the neurotransmitter is put out into the synaptic cleft. Now here, during exocytosis, some part of the membrane is added on to the neuronal membrane. It is retrieved back by endocytosis. That we shall see in the next diagram. That part will become clearer. Now, this is not just the only mechanism. There is one more mechanism proposed wherein the vesicle just fuses with the neuronal membrane and a small hole like is punched in the membrane. It releases the contents and it rapidly seals off, which means the vesicle stays within the cell. It doesn't have to add on its membrane to the neuronal membrane. So this has been called as charge. So the vesicle just comes and kisses or is near this membrane, and the contents are put out via a small hole which is produced, and it rapidly reseals. So this is known as kiss and run discharge. To make things more clearer, now this diagram, this is taken from textbook of Ganong. Here you can see a neurotransmitter vesicle, synaptic vesicle. This comes and docks near specific areas of the neuronal membrane, which are called active sites. So the synaptic vesicle comes and docks here. And there is something called priming occurring. A priming actually enables the synaptic vesicle to rapidly. See, whenever an action potential reaches, there should be fast activity. It should readily release the neurotransmitters. For this, some proteins will prime the synaptic vesicle. And when the membrane potential, the action potential reaches the terminal ending, the voltage change occurs. Then it leads to calcium influx via the voltage gated calcium channels. This calcium leads to exocytosis of the neurotransmitter into the synaptic cleft. So here there is fusion of the synaptic vesicle membrane, and then only the area of fusion is breaking down, the neurotransmitter is being released. So what will happen to the remaining membrane which is added on to the neuronal membrane here? A protein clathrin comes and coats this vesicle, and finally this is endocytosed or taken back. So a combination of exocytosis and then endocytosis retrieves back the synaptic vesicle once it has performed its action of releasing the neurotransmitter acetylcholine. Are the various steps clear? At least you should know what is docking, that exocytosis is occurring. For it, you require the opening up of voltage gated calcium channels. Is this much clear? Any doubts that you have in this? Now, there are a lot of other changes also which are occurring. As calcium influx occurs, this calcium is sensed by a protein which is called synaptotagmin. Now here there are some different names that you are going to learn. There is a synaptic vesicle associated calcium binding protein called synaptotagmin. So entry of calcium or the calcium signal is sensed by this protein which is called SNARE. Now don't be scared by the expansion of SNARE. It is soluble NSF attachment protein receptor, where NSF stands for N-ethyl maleimide sensitive factor, or simply know it as snare protein complex. So calcium is sensed by a vesicle associated calcium binding protein called synaptotagmin that activates snare proteins. Now snare is actually a complex. There are three main structures involved here. And the three are synaptobrevin, syntaxin, and SNAP25. Now, whenever the names become difficult like this, it gains importance from point of exam. Now, synaptobrevin, it is also known as V-snare, a protein which is present on the synaptic vesicle. It is present in the vesicular membrane. Syntaxin, it is present in the target membrane. In our case, is a muscle. It is also known as T-snare. So it's easier to know it as synaptobrevin as V-snare and syntaxin as the target or T-snare. There is another protein which is SNAP25. The expansion of it is synaptosomal nerve-associated protein 25. And there are many few associated proteins with it, but at least these three we should be knowing. Now, we shall see where these proteins are present. What do you mean by V-snare and T-snare? Where are they located? This we shall see in a diagram. Now, this is a diagram taken from textbook of Ganong. This part shows the synaptic vesicle membrane, and here is the synaptic cleft, and next part is the membrane of the target. Now, the first component that we discussed as V-snare or vesicular membrane, the protein synaptobrevin, it is shown here along with the vesicle. Yes, somebody's mic is on. If you have any doubts, you can ask. Okay. So on the synaptic vesicle, you have synaptobrevin. It is a T-snare protein called syntaxin. I hope the location of here in this diagram. And along with this comes SNAP25, which is synaptosomal nerve-associated protein 25. This is also known as a ternary complex where three components are present. So keep in mind, synaptobrevin or V-snare is present on the synaptic vesicle. Syntaxin or T-snare is present on the target membrane. And associated with this, you have SNAP25. There is NSF. We have heard this name before. N-ethyl maleimide sensitive factor. And this RAP3, it is actually small GTPase. So diagram itself shows you the complexity with association of the proteins at the synapse as the synaptic vesicles come and dock, and then only they release the contents. Are the components in the diagram clear? Any doubts that you have? Any doubts that you have?
Ma'am, conduction target membrane-ലാണോ അതോ അതിന്റെ between-ലാണോ ഇരിക്കുന്നത്?
Clear ആയില്ല, ഒന്നുകൂടെ ചോദിച്ചോളൂ.
Syntaxin target membrane-ലാണോ അതോ synaptic vesicle-ന്റെ between-ലാണോ ഇരിക്കുന്നത്?
Synaptic vesicle ഉള്ളത് V-snare ആണ്. That is synaptobrevin. And syntaxin is on the target membrane. And these two are coming and interacting along with SNAP25. You also require some added proteins like this MUNC and also NSF. All these together only help you to have the integrity of the synapse.
Okay, ma'am. Thank you.
Okay. Is your doubt clearer?
Yes, ma'am.
Okay. I hope the screen is still visible. Yes, somebody's mic is on. Any doubts that you have, you should be asking, not otherwise. Yes, I hope you can see the screen now.
Yes, ma'am.
So after describing the presynaptic component, we shall move on to the second component, which is synaptic cleft. Now the synaptic cleft, as you know, it's a small gap that you have between the presynaptic and the postsynaptic components. Now if it is a neuromuscular junction, it's between the nerve, presynaptic neuron terminal, and a muscle part, which is motor end plate, that we shall discuss after discussing the synaptic cleft. Now, how small is the synaptic cleft or the gap which is present at the synapse? Mentioned as 20 to 40 nanometers. The reference for this is Ganon, or your textbook of Guyton mentions it as 40 or 50 to 100 nanometer. Now, this is something which might come as a difficulty as you read your textbook, but one of the standard values you can follow. So 20 to 40 nanometer, that is the normal width of the synaptic cleft. Now the neurotransmitter that was released from the presynaptic nerve terminal by exocytosis, it enters the synaptic cleft. This neurotransmitter is acetylcholine, and this diffuses across the synaptic cleft. Now this diagram shows a presynaptic neuron, some areas at the synaptic terminal or the synaptic knob called active zones, where the synaptic vesicles come nearer. They are going to come and dock there and stay ready to release the contents by exocytosis. In the postsynaptic cell, there is an area called postsynaptic density, which contains the receptor proteins. Now, this is the area where the neurotransmitters are going to come and bind. The postsynaptic part that we are going to discuss in a neuromuscular junction, it is the part of the muscle. Now, that part of the muscle which takes part in the synapse here, it's known as the motor end plate or the end plate membrane. Now, when a nerve is forming a synapse with a muscle fiber, it comes and ends at the center of the muscle fiber. So the advantage here is that once that center part is going to be stimulated, and if an action potential is generated, it is going to be propagated in both the directions to both ends of the muscle fiber. So here, this is a new term that you have to keep in mind in a neuromuscular junction. The presynaptic neuron's terminal button is in close approximation to the part of the muscle which is called motor end plate. Now, this motor end plate has a special feature. The membrane is thrown up into multiple folds, and these folds are called junctional folds. So these folds are going to contain the receptor for acetylcholine. So what is the advantage of having so many folds? Literally, it increases the surface area which is available. So more receptors can be accommodated here. So more neurotransmitter acetylcholine can come and bind. So the key information here, in the muscle, you are having a motor end plate with a lot of junctional folds. These contain the receptors where the neurotransmitter acetylcholine is going to bind. Now it is quite surprising. How is it that there is a gap between the presynaptic and postsynaptic membrane, but still the synapse is maintained as such, and specifically that neurotransmitter is released and it comes and binds there? Now the answer actually lies in proteins. There are proteins called neurexins which are present in the presynaptic membrane, and they are going to interact with neurexins on the postsynaptic membrane. They actually hold the synapse, maintain its integrity, and also provide a mechanism for the specificity of the synapse. So neurexins, this is just good to know. The name neurexins. That we mentioned on the junctional folds which are present in the motor end plate. The receptors for acetylcholine in the muscle, these are of nicotinic type, and these are called nicotinic acetylcholine receptors. This is the accepted short form in which it is written. Ligated channel, which means some ligand or substance has to come and bind, then only this channel opens. The neurotransmitter acetylcholine, when it comes and binds to the receptor, it opens a gate and then sodium influx occurs. So just refresh again. If sodium is going to enter into the membrane of the muscle, what will happen to the membrane potential there? Initially, the membrane was in the polarized state. It was having a resting membrane potential. And now sodium enters, interior becomes positive, the normal polarity is lost, or depolarization occurs. So almost 70 millivolt depolarization can occur. And these small potentials, which are generated at the motor end plate, these are called end plate potentials. Now, if you just recollect from the nerve action potential, how much of depolarization do you require to reach up to the firing level? I'm asking you, from a neuron resting membrane potential of minus 70 millivolt, if you have to reach up to minus 55 millivolt, what is the magnitude of depolarization required? You require just around 15 millivolt, isn't it? That is quite small. Now, similarly, even for a muscle, you require only around 15 millivolt depolarization, but then this neurotransmitter acetylcholine, when it opens up voltage gated sodium channels and the sodium is entering, ligand gated channels where acetylcholine is binding, these sodium ions, as they enter, they are causing a large amount of depolarization, almost 10 times of what is required. This is actually a safety factor, so that when an information via the nerve, it is coming as an action potential, it should cause a change in the membrane potential and lead on to development of action potential. So this end plate potential, which is having the capacity to take the membrane potential to the firing level, this then leads on to the action potential. Now the action potential which is developed in the muscle will be dealt later on along with the muscle component of the lectures. For now, what you have to keep in mind, an action potential, when it reaches the presynaptic neuron, causes acetylcholine release. The acetylcholine binds to the nicotinic type of acetylcholine receptors on the motor end plate. This leads to sodium influx, depolarization occurs, and there are potentials generated called end plate potentials. If they take the membrane potential to the firing level, there is an explosive opening up of voltage gated sodium channels leading to action potential. And once generated, the action potential propagates in both directions. So this is just to give an idea of how a ligand comes and binds and then leads to sodium influx, leading to interior depolarization. This we are talking about the postsynaptic cell, which here it is a muscle. The synaptic vesicles These are spherical structures. These are also known as quanta or packets of acetylcholine. Why do we call them as quanta? These packets, they contain neurotransmitters. Now, normally, even in state, small quanta of acetylcholine are released to cause small changes in the enclave potentials. And their magnitude is quite low. Those are called miniature enclave potentials. When an actual action potential reaches, it leads to release of more quanta or packets of acetylcholine. But then each action potential coming leads on to release of the same amount of synaptic vesicles. So each action potential leads to around 60 vesicles to release the neurotransmitter, and each of these vesicles contains around 10,000 acetylcholine molecules. So here also, as a quantity, there is excess of neurotransmitter being released. This also is a safety factor which allows to ensure that action potential does occur. So more than what is required, that much acetylcholine is going to be released. Now this diagram, it is taken from the textbook of Guyton. This is the classical diagram of a neuromuscular junction. See the first component, which is a presynaptic neuron, its terminal ending, which is enlarged form. What is this structure called? The terminal enlarged part. The synaptic knob or the synaptic button. And in red, there are some areas shown which are the active zones. Here, the synaptic vesicles come nearer and they stay here. They will dock. They will exocytose the contents into the synaptic vesicle. Membrane sarcolemma, it is thrown into extensive folds where the synapse is being formed, and this area is called motor end plate, the part of the muscle where the neuromuscular junction is formed. This is called the motor end plate. Here there are a lot of junctional folds. These folds contain the receptors for the acetylcholine. The nicotinic acetylcholine receptors are present here. Are the different components of the diagram clear? First component, presynaptic neuron. Second component, synaptic cleft. Third component, it is the motor end plate of the muscle. Any doubts that you have here till now? Now this diagram, you should be able to draw, labeling all the important components. If you take, this is another diagram from Guyton itself, which shows the events which are occurring sequentially numbered. Number 1, from the motor neuron, action potential reaches the terminal nerve ending. Number 2, the voltage change causes opening up of the voltage gated calcium channels. So calcium influx occurs. Number 3, this causes exocytosis. Neurotransmitter acetylcholine is released. Where will acetylcholine come and bind? Somebody's mic was on. Now they realized it very fast. That is good. Are you able to see the screen?
Yes, ma'am.
Okay. So we were just discussing the events occurring at the neuromuscular junction. We are just revising the events that we have discussed till now. Number 3, as acetylcholine is released into the synaptic cleft, it diffuses and it comes and binds to the nicotinic cholinergic receptor for acetylcholine. Number 4, ligand-gated channel leads on to opening up and sodium enters. This sodium leads to depolarization of the membrane, the motor end plate. Small potential changes develop here. The local currents are developed. These potential changes are called end plate potentials. They summate. Now, this change is being transmitted across the sarcolemma. If the end plate potentials take the membrane potential to the firing level, this will lead on to explosive opening up of voltage-gated sodium channels. Sodium influx will occur. Action potential will be generated in the muscle. This actually summarizes the events occurring at the neuromuscular junction. Do you need a small break? Or shall I continue?
A small break.
Maybe 5 minutes. Now it is 10 o'clock. We shall continue by 10:05.
Yes, ma'am.
In the meantime,
Okay,
I'll play a small video.
The schedule, there is a class at 10 o'clock.
Muscles.
It is good to notice that. Well, I'll be taking till 10:25. Please remind me at 10:25. After that, Dr. Sneha will be taking. Is that okay? It's good to. So the neuromuscular junction.
Ma'am, it's not clear.
Ma'am, audio is not clear.
Okay, the volume is not clear. Can identify the two components: the presynaptic neuron, the terminal knob, the synaptic cleft, and the motor end plate with the junctional folds. You can see the nicotinic cholinergic receptors. Now, when an action potential comes down the neuron and the synaptic vesicles release neurotransmitter acetylcholine, it comes and binds to the nicotinic cholinergic receptors, which is a ligand-gated channel that causes sodium influx. Now here, on the top part, you can see this causes a small increase or depolarization in the membrane potential. If it reaches the firing level or threshold, then only the further changes occur. The end plate potential, it should be able to lead on to further sodium influx. The voltage-gated sodium channels open and a rapid increase in the muscle membrane potential occurs. That's how an action potential will be generated in the muscle. To give an idea how the sequence of events occur, and as I mentioned, the motor nerve ends on the mid portion of the muscle fiber. Therefore, once an impulse is generated, it can spread in both directions. Yes.
Break
For you.
അപ്പൊ 2 മണിക്കൂറിന് ആയിരിക്കും 1 മണിക്കൂർ മാർക്സിൽ 11 മണി വരെ അല്ല 12 വരെ
Short break over. Exactly 5 minutes, no? Okay, are you able to see the screen?
Yes, ma'am.
Yes, ma'am.
So we just discussed the neuromuscular junction and how transmission is occurring across it. So then comes a natural query. Once a nerve impulse is transmitted to muscle and the muscle responds by generating an action potential, how is the signal then terminated? The signal is terminated by degradation of the neurotransmitter acetylcholine by acetylcholine esterase. This is present associated with both the membranes, presynaptic and postsynaptic. Another fate of the neurotransmitter, it can be recycled. It can be taken up by the neuronal terminals or even some glia. Actively, it is taken up. And again, it is recycled and repackaged into synaptic vesicles. So this recycling by reuptake actually helps in maintaining a supply of the neurotransmitter. It may just affect the neighboring neurons. But as such, in that synapse, the amount of neurotransmitter being reduced, the signal can be terminated. That's the usual fate of the transmission. Okay. Now coming to acetylcholine esterase, the enzyme, which actually hydrolyzes acetylcholine to two components, acetate and choline. What happens to acetate? It just diffuses into the extracellular fluid, whereas choline, it is taken up again. There is reuptake of choline at the nerve terminals. So we'll just have a look at a diagram which just briefs these changes. That will help you to understand the changes better. Now, this is a presynaptic nerve terminal which is shown to just get an idea of synthesis of acetylcholine. It is acetyl-CoA and choline which unite in the presence of choline acetyl transferase to synthesize acetylcholine. Once the acetylcholine is packaged into synaptic vesicles and it enters into the synaptic cleft by exocytosis. Just a mention that there is a transporter protein required for entry of this acetylcholine into the synaptic vesicles. It's called vesicular acetylcholine transporter. So a lot of proteins are involved in the transmission. Choline is exocytosed into the synaptic cleft. It binds to the receptor and it produces its action. Now, to terminate its action on the nicotinic cholinergic receptors, the acetylcholine has to be degraded or hydrolyzed by acetylcholine esterase. We had mentioned that choline, it can be taken up back and then it is reutilized and recycled during the synthesis of neurotransmitter acetylcholine. So this cycle keeps operating to maintain a supply of the neurotransmitter acetylcholine. So the diagram is just to show you how the reuptake is occurring, how it helps in the synthesis of further acetylcholine. I hope that part is clear. Just a brief mention of the neuromuscular blockers. Now, just like in a synapse, you can have presynaptic blockers, also postsynaptic blockers. Presynaptic blockers, one of the important things to mention is botulinum toxin, a toxin which is produced by the Clostridium botulinum bacteria. Now the botulinum toxin B, F and G, they inactivate a protein called synaptobrevin. Now just try to recollect, where is this synaptobrevin present? A clue here. The V I have shown in color. Why? It's present in the synaptic vesicular membrane. Yes. So the botulinum, these toxins, they are inactivating synaptobrevin. So the synaptic vesicle cannot fuse. If there is no fusion, there can't be exocytosis also. So neurotransmitter acetylcholine cannot be released. So this is a sort of presynaptic block of transmission. If it is botulinum toxin A and B, they act on SNAP25. If it is botulinum toxin C, it breaks down syntaxin. Now that is the protein which is present on the target membrane. Now this shows different toxins are acting on different components of the snare complex proteins. So if any problem occurs here in the synaptic vesicles fusion with the membrane, release of acetylcholine cannot occur, and therefore the effects thereafter will not be seen. Now, typically, this type of presynaptic blockage by botulinum toxin, it causes flaccid type of muscle paralysis in cases of botulism, where there is poisoning due to excess production of this toxin. Now, any use to this botulinum toxin that many of you will be knowing? Any use of this toxin? Somehow it wasn't clear. Yes, at least a cosmetic use you will be having an idea, just being used by those in the media profession. The cosmetic use, injection botox, can be used to remove wrinkles. Now, botulinum toxin, it is having therapeutic or treatment purpose. Its use is as a muscle relaxant, especially in conditions like echalasia cardia, where in the lower esophageal sphincter, it is tonically contracted, and to cause relaxation there, it can be injected at that site and causes relaxation of the muscle. Or sometimes as part of treatment for strabismus or squint, it may be injected to certain extraocular muscle to cause relaxation of that muscle. And the cosmetic use, where injection botox, injection into the facial muscles causes relaxation there and reduces the aging effects or wrinkles. So better appearance. That is what is being used by the film stars. By hemipolynia. Now this inhibits the reuptake of choline at the presynaptic level. Therefore, there can be neurotransmitter depletion, so action potential will not occur. So there are different mechanisms for presynaptic block. Now considering...
Ma'am, previous slide on the
Yes, sure. What is more important is botulinum toxin.
Thank you.
Okay. Postsynaptic block. Here we are referring to any interference with end plate potential, and therefore there is interference with generation of action potential. There are two types of postsynaptic blocks that can occur. The first type is competitive blocking. Now there are some competitive blockers. They compete with acetylcholine and bind to its receptor. Which is the receptor for acetylcholine? It is a nicotinic cholinergic receptor. Say for example, they bind to the nicotinic cholinergic receptors. Therefore, acetylcholine cannot exert its action. So this is a type of competitive blockade where Since neurotransmitter cannot bind to its receptor, action is not seen and there is blockage of transmission. Now the second type of postsynaptic blockers, these are depolarizing blockers. Now these act just like acetylcholine, but they are not destroyed by the acetylcholine esterase enzyme, which means acetylcholine-like action is going to remain for a longer time, and therefore the muscle remains depolarized or stimulated for a longer time. So action just like acetylcholine, so it is a stimulatory effect or depolarizing block. So muscle spasms occur, effective muscle contractions cannot occur. Example of two such blockers are succinylcholine and carbamylcholine. So they cause more action of acetylcholine, so more of depolarization of the membrane. Therefore, these are known as depolarizing blockers. Little bit confusing. Now, there is also another type of inactivation where there is inactivation of the enzyme acetylcholine esterase. Now, this enzyme is actually hydrolyzing acetylcholine. So, if there is inactivation of acetylcholine esterase, what will happen to the amount of acetylcholine? It increases. So, more amount of neurotransmitter acetylcholine, more stimulation occurs at the neuromuscular junction. Can be of two types, reversible type and irreversible type. The reversible acetylcholine esterase inhibitors, for example, neostigmine, physostigmine, these are used as part of treatment for a disease, Myasthenia Gravis. Have you heard the name Myasthenia Gravis? It's an autoimmune disease where specifically there is destruction of receptors in the muscle membrane. The nicotinic cholinergic receptors are destroyed. So here is a situation where whatever some functional receptors are present, if you are giving these reversible AC inhibitors, they will not destroy whatever acetylcholine is available, and therefore those neurotransmitters can produce their effects. So this reversible type of AC inhibitors are used to improve the muscle activity in myasthenia gravis. Name one film personality who has myasthenia gravis. A good actor who has myasthenia gravis. Generally, everybody gives the answer. It's Amitabh Bachchan. Even he has myasthenia gravis. Acetylcholine esterase inhibitors, which are of irreversible type. This is seen in cases of organophosphorus poisoning. Say, for example, with parathion, malathion, or bagon, or certain nerve gases like sarin or diisopropyl chlorophosphate. These are used in biological warfare. They cause muscle paralysis. Somebody's mic is on. Okay. Any doubts in what is taken till now? Shall I continue with little more about nerve injury?
Okay, ma'am.
Now, one thing that you can do is attendance. So instead, on Wednesday, those who attend can the diagram of neuromuscular junction. So on one page, the diagram of neuromuscular junction. On the other side, action potential of neuron that all of you can submit. Yes. Now, you are able to see the screen?
Yes, ma'am.
Okay. Now, your next specific learning objective is to know regarding peripheral nerve injury, what is meant by Wallerian degeneration, few things regarding regeneration, and also what is denervation hypersensitivity. Now, this is something Wallerian degeneration has often been asked for exams, right, by different people. But Sunderland's grading is what we are going to discuss. It shows 5 degrees or types of nerve injury, where the first degree of nerve injury is due to mild pressure on the nerve. It leads only to a temporary or transient loss of function due to local ischemia, lack of blood supply, and thereafter complete recovery occurs. The second degree nerve injury, in situations where there is prolonged pressure on the nerve, but the endoneurium, the innermost connective tissue layer, remains intact. In such cases, recovery may be delayed, but still complete regeneration of the nerve can occur. So these are the 2 degrees where recovery, there is not much of a problem. Now for 3rd degree, 4th degree, and 5th degree of nerve injury, there is more severe damage. In the 3rd degree of nerve injury, severe damage interrupts the endoneurium. If there is also disorganization of bundles of the nerve fibers, that is fasciculi, it is called 4th degree. If there is complete transection, so through and through cutting of the nerve, which is called 5th degree nerve injury, in such cases, recovery will not be complete. And whatever some recovery occurs in the 3rd and 4th degree takes very long time. To Wallerian or orthograde or anterograde degeneration, whenever there is a injury, the part distal to the cut segment shows some degenerative changes. Those changes are called Wallerian degeneration or anterograde degeneration. So wherever the cut has occurred, from there, the terminal ending part of the neuron is undergoing some degenerative changes. The first change that you can see, synaptic transmission stops because there is an injury. The axon generates. The myelin sheath breaks down, and some round fatty droplets like beads are seen, which are of cholesterol ester. Now, this is a diagram in which you can see a neuron. Now, if there is a cut injury, the distal part undergoes degeneration, which is called orthograde or Wallerian or degeneration. Now, if injury changes are proceeding in the reverse direction, we call this as retrograde degeneration. Somebody's mic is creating this.
Ma'am, it's 10:25.
Yes, it's time, so I'll be stopping now. There are some few more things to be taken. I'll see how I can take it. I might upload the class depending upon the time available. So please read up and please submit what is asked on Wednesday. So Wednesday, refractory period, just a single page I want. I don't want you to write long essay. Diagram of neuromuscular junction, diagram of neuron action potential. So just 2 pages. Write your name, roll number also. So please keep reading. Any doubts you can ask.
Okay, ma'am. Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
ഫോണിന്ന് കയറാൻ നോക്കി. Am I audible to all of you?
Yes,
Good morning,
ma'am.
everyone.
Good morning, ma'am.
Okay.
Good morning.
Morning. So we have a very small portion left behind in blood grouping. Have you all studied about the ABO system, RH system, erythroblastosis fetalis? How will you manage a case of erythroblastosis fetalis and all? Padicho? Padicho? So who is going to tell me how will you manage a case of erythroblastosis fetalis? How will you prevent erythroblastosis fetalis? How will you prevent erythroblastosis fetalis?
Anti-RH
By injecting anti-D serum.
Yes, very good. Very good. Within how many hours will you give anti-D?
72
hours. How does it act? How does anti-D act?
It
Destroy
destroys
the RH positive cells in mother and prevent.
Yeah, it by two mechanisms. One is the cells which has entered, the fetal cells which has entered in the maternal circulation will be neutralized by the antibodies, and also it prevents the sensitization and formation of further antibodies by the mother. Okay, so we have talked about RH incompatibility, no? So, and I have told you that incompatibility, whether RH or ABO incompatibility, that occurs in two situations. Which are the two situations? ABO incompatibility ആണെങ്കിലും RH incompatibility ആണെങ്കിലും രണ്ട് അവസ്ഥകളിലാണ് ഉണ്ടാവണേ. എപ്പോഴാണ് ഉണ്ടാവണേ? One is in mismatched blood transfusion. അല്ലെ, തെറ്റായിട്ടുള്ള ബ്ലഡ് നമുക്ക് ട്രാൻസ്ഫ്യൂസ് ചെയ്ത് കഴിയുമ്പോഴത്തേക്കാണ് നമുക്ക് ABO അല്ലെങ്കിൽ RH incompatibility ഉണ്ടാകാവുന്ന ഒരു കണ്ടീഷൻ. Right? You will get ABO incompatibility or RH incompatibility when you are being transfused with the wrong kind of blood, right?
Yes,
ma'am.
Okay. And we have explained another condition where you can get RH incompatibility. Which was that?
Hemolytic
Disease of newborn. So shall I tell you something else? I mean, something very interesting.
RH negative.
Yes, even though we have discussed about RH incompatibility, we have one more thing. What is that called as? Hemolytic disease of newborn can be due to, not only due to RH incompatibility, it can also be due to, ABO incompatibility. Can you see my slides? Can you see the slides?
Yes, ma'am.
Yes, ma'am.
Okay.
Yes.
What are you seeing there on the slide? എന്താ കാണണേ?
Zoom meeting and
Zoom.
dialog box.
Zoom sign in there. The box.
ഒന്നും മനസ്സിലാവുന്നില്ലല്ലോ.
Zoom is
able to see the screen of ABO incompatibility screen?
Yes, ma'am.
Okay. So why I have discussed a lot about RH incompatibility and I am discussing about ABO incompatibility after that? എന്തുകൊണ്ടാണ് ഞാൻ ആദ്യം ABO incompatibility പറ്റി ഒന്നും പറയാതിരുന്നത്? പറ. Why?
കണ്ടുപിടിക്കാൻ എളുപ്പമാണ്.
കണ്ടുപിടിക്കാൻ എളുപ്പമല്ല, because it is not of much clinical significance. It won't cause much clinically എന്താ പറയുക significant ആയ issue ഉണ്ടാക്കത്തില്ല. It's a very less severe condition. ABO incompatibility, it's a very common condition, but it is not of much severity. When will you get ABO incompatibility? You think in terms of RH. You will get, can you see my screen here?
Yes, ma'am.
Can you see a picture here? So see, this mother is pregnant. She is of blood group O group. So has she got any antigen on her red cell surface? Has she got any antigen on her red cell surface? O blood group.
No.
And what about the antibody? What about the antibody?
Antibody.
What is the nature of the antibody? What type of antibody is that? Normally IgM. But there can be, yes, IgM is the type of antibody, but there can be situations where you can have IgG type of antibody also. Clear? Is that clear? The most common type is IgM, but there can be IgG type of antibody. So what this antibody can do? If the baby is, yes, it can cross the placenta. So if the baby is A or B blood group, the mother will be having A antibody or B antibody, which is of IgG type. It may cross the placenta and it may reach the fetal circulation and it can cause hemolysis in the baby. But usually it occurs in a very small amount. It won't cause much of reactions and usually it will be mild. And usually it manifests in first pregnancy. In RH incompatibility, which pregnancy was affected? Is it the
Second pregnancy.
Second pregnancy. First pregnancy usually spares. Unlike that, here it is the first pregnancy usually affected. In the second pregnancy onwards, it won't cause much of the reaction. Is that clear?
Yes.
Is that clear? Okay. So for completion's sake, I have been telling you that. So the hemolytic disease of newborn can be either due to RH incompatibility or due to ABO incompatibility. But the clinically significant one is the RH incompatibility, which occurs in a mother who is RH negative and the baby who is Rh positive. Okay, and the antibody of Rh system is IgG type. Okay, and we have seen how you can manage a case of Rh incompatibility. We have seen how you can prevent the condition and how you can treat the condition. What are the treatment options available for managing erythroblastosis fetalis? How will you treat? Exchange transfusion. Don't say that blood transfusion. It is called as exchange transfusion. And what is the other option?
Phototherapy.
Phototherapy. What is the principle behind phototherapy? Bilirubin is converted
Photo-oxidation of
into
bilirubin.
lumibilirubin, which is a more soluble form.
Soluble.
Okay, so the differences from Rh incompatibility, this occurs in usually in first pregnancy, less severe, it requires no treatment. Rh incompatibility I have discussed first and then I have discussed the ABO incompatibility. So we have seen what is called as the hemolytic disease of newborn. Okay, now what I am going to tell you is what you will do in your for your hematology practicals. Practical that is blood grouping. How many of you don't know your blood group? Is there anyone who don't know your blood group? Is there anyone who don't know your blood group? Blood group ariyatha aarengilum undo? Is there anyone who don't know your blood group? Anyone? Is there anyone? So can anyone please tell me how your blood group was found out? How was it found out?
Baby card.
Baby card. Okay, very good. Baby card. Okay. Then, for those who this baby, this typing was not done at the time of birth. What was the method? You might have went to a lab. You might have went to a lab. It's a very simple procedure. You will take out. I'll tell you what you will do in the lab when you come for the practicals. You will clean your finger with spirit. You will allow it to dry. Then you will prick your finger. You will discard the first drop of blood. Then you will add the second and third drop of blood. To that, you will take that blood in a watch glass. Can you see a watch glass here? Can you see a watch glass here? Glass under chemistry lab practical biology dicot monocot.
Yes, ma'am.
Dicot monocot. So you will take watch glass, right? Are you able to hear me?
Yes, ma'am.
Okay. So what will you do there? You will take out thin sections of. Can you hear me?
Yes, ma'am.
Yes.
Okay. Solution dicot monocot stem watch glass.
Yes, ma'am.
Okay. Watch glass you will add blood by finger puncture method, and to that you will add isotonic saline, citrated isotonic saline. Is that clear? Is that clear?
Yes, ma'am.
Okay, then you will make a suspension of the red cell. Where blood saline mix it, you will make a suspension. Then what will you do? You will take four glass slides. In the lab, we will provide you with 4 glass slides. To one slide, you will add what? You will add anti-A serum. Can you look? Can you see this picture? Can you see this picture? Can you see the slide?
Yes, ma'am.
Yes, ma'am.
Okay. Yes. So what is provided here is actually this blue colored thing is the anti-A serum. Can you look? Can you appreciate the color of anti-A serum? What is the color? Anti-A serum. What is the color? It is blue in color. It is blue in color. Okay. So you will take or you will keep a drop of anti-A serum on the glass slide, and you will add a drop of blood into that. Okay, then what will you do? You will take anti-B serum. What is the color of anti-B serum? No, Kike, what is the color?
Yellow.
What is the color of yellow in color? You will take a drop of anti-B serum also in another slide, and into that also you will add a drop of blood. And to another glass slide, you will add anti-D serum. What is that? D serum. What is the color? What is the color?
Colorless.
It is colorless. Colorless. And we will also keep one slide as control. You will simply add isotonic saline to cross check. Endingle errors to see whether any mistake or any error has happened in your method. You will keep one control also. Are you able to appreciate that this sera being placed on the glass slide here? Are you able to see that? Are you able to appreciate that? Actually, this has been placed on a single slide, but for us, we will keep separate slides for each one. We'll give you separate slides. So to one slide, you will place anti-A sera. To another slide, you will place anti-B sera. To another one, you will place anti-D sera. And to one, you will place isotonic saline as a control. Then what will you do? You will add, you have already prepared red cell suspension. No, you have mixed blood with saline and you have prepared red cell suspension. To each of these anti-sera, you will add a drop of red cell suspension. Is that clear?
Yes. Yes, ma'am.
Are you able to follow me? Okay. So we'll see what happened. So see, this is a person who is having type A. Okay, type A blood group individual. What has happened here with anti-A blue colored? What has happened here?
Agglutination.
Yes, very good. That process is called as, what is it called as? Agglutination. That is the term. What is that called as? It is called as agglutination. So this antibody A containing sera has reacted with cell which has got antigen, dash on its surface. A antigen A antigen on its surface. Is that clear? Is that clear? And what has happened with anti-B? Is there any B antigen on the red cell surface for A blood group individual?
No.
No. So has any agglutination occurred here? Is there any agglutination here with anti-B?
No, ma'am.
Anti-B യുടെ കൂടെ എന്തെങ്കിലും agglutination നടന്നിട്ടുണ്ടോ? No. You look at type B. Type B, what has happened? Type B with anti-B, what has happened? What can you see here? This is, can you see the clumps?
Yes,
Clump കണ്ടോ? ചെറിയ കട്ടയായിട്ട് കിടക്കുന്ന കണ്ടോ? ഒഴുകിയാണോ നടക്കുന്നത്? ക്ലിയർ ആയിട്ടാണോ ഇരിക്കുന്നത്? അല്ല, കട്ടയായിട്ടല്ലേ ഇരിക്കുന്നത് ഇവിടെ റെഡ് സെൽസ്. What is that called as? It is agglutination. So if the individual is type B blood group individual, agglutination will occur with anti-B serum, but with anti-A serum, no agglutination will occur. But if the individual is AB type of individual, agglutination will occur with both A and both B. Is that clear? Because the AB type of individual has got both A antigen and B antigen on the surface. So it will react with both A antibody and B antibody. Is that clear? Clear?
Yes, ma'am.
Yes, ma'am.
അയ്യോ, ഇതാരാ സ്ക്രീനിൽ ഇങ്ങനെ ആയിട്ട് മീൻസ് എന്തൊക്കെയോ കുറെ എഴുതുന്നുണ്ടല്ലോ. So how will you recognize whether it is agglutination or not? നമ്മുടെ ആ slide, you can gently shake the slide. Okay, so if it is agglutination, the cells will not separate. It will form, actually you call it as red chili appearance, chili powder appearance. Okay, so sometimes you may get a confused feel whether this is agglutination or not. So in that case, you can just simply you can just move the slide, and still if the cells are having that chili powder appearance, if it forms that clumps, if it is not getting dissolved, that means it is agglutination. If you still have doubt, if you are doubtful whether this is agglutination or not, what can you do? You can look it under microscope. Clear? If you have doubt whether this is agglutination or not, what can you do? You can look it under microscope. Okay. Then a word about forward typing and reverse typing. So what I've mentioned right here is the forward typing, where the cells are mixed with the source of anti-sera or antibody. What is reverse typing? What is reverse typing? What is reverse typing? We will take out the sera of the recipient or patient. We will take the sera of the patient. Then you will add group A and group B blood cells or red cells into it. Are you able to follow me? Are you able to follow me? What is reverse typing? മനസ്സിലാവുന്നുണ്ടോ ഞാൻ പറഞ്ഞത്? Forward typing നമ്മൾ എന്താ ചെയ്തത്? You have mixed the patient's red cells with anti-sera. But in reverse typing, what you will do is you will take the sera of the patient or plasma of the patient, serum of the patient, and you will mix it with the commercial source of group A and group B red cell. Is that clear? Is that clear?
Yes.
So you look at this picture. If it is not clear for you, look at this picture. This is the sera of a patient who is having AB blood group. Okay, so you have taken sera into test tube, and to one sera you have added A blood cell, and to another sera you have added B red cell. So in AB blood group individual, has he got any antibody in the plasma? AB individual, has he got any antibody in the plasma?
No.
So there will be no antibody in the plasma. So if you add antigens into the test tube, will it cause agglutination? Will it cause agglutination? No. Is that clear? Is that clear?
Yes, ma'am.
Is that clear?
Yes, ma'am.
Okay. So what about B? What about B? So you have anti-A sera. Sorry, you have sera taken in 2 test tubes. To one you have added A red cell, and to another you have added B red cell. So what has happened here? What has happened here? നോക്കൂ, B blood group individuals ഏത് ആന്റിബോഡിയാ ഉള്ളത്?
Anti-A.
So that anti-A will react with A antigens. Clear?
Yes.
A antigens. So you will get agglutination here. Similarly, for A blood group, you will agglutination with B red cell. If it is O, they have got both A antibody and B antibody. So they will react with both A red cells and B red cells. So that process is called as reverse typing or serum typing. Is that process, is that explanation clear for all of you? Is that clear for all of you?
Yes, ma'am.
Yes, ma'am.
Okay. So So now comes the media that enhances agglutination. So if you have added albumin or enzymes like papain, trypsin, etc., it will increase the sensitivity of the antibodies, particularly of certain blood groups like Rh system. And if you decrease the ionic strength of the solution, that also can enhance the process of agglutination. Clear? So comes the most important part. What is the importance of blood grouping? What is the importance of blood grouping? Why you want to do the blood grouping?
The transfusion.
Yes, most important and the most common use of blood grouping is for blood transfusion, right? Right?
Yes, ma'am.
Okay, blood transfusion samayathalle nammal usually you are doing the blood grouping, right?
Yes, ma'am.
Okay, and also for organ transplantation, you might have seen, no, people who are in need of organ transplantation, A blood group ulla kidney ulla aale avashyamund, B blood group angante parasyam kanditille?
Yes, ma'am.
Okay, so for organ transplantation also the blood group is very, very important. And to solve disputed paternity cases, for example, if suppose if the baby is of AB blood group, and if someone says that the O blood group individual is accused of being the father, and if the mother is of A blood group, you can't say that this person is the this person will be the father. So if this person won't be the father, you can say with blood grouping. Is that clear?
Yes, ma'am.
Okay. And also another case situation is from the blood stains which are obtained from the site of crime. Okay, so from the site of crime, suppose if the accused person, the blood stain obtained from the site of crime and also that accused person is the same, then you can say that this person can be the culprit. Okay, and also in pregnancy to avoid Rh incompatibility. And another thing is some disease, it is more common in people who are of a particular group. For example, duodenal ulcers are more common with O group individuals. Gastric carcinoma and pernicious anemia are common with A blood group individuals. And urinary tract infections are common in females with B and AB blood group. So that is the importance of blood group. So what all things are the importance? So one is to prevent mismatched blood transfusion and organ transplantation, to solve disputed paternity cases, and to identify accused persons from blood stains that are obtained from the site of crime, and in pregnancy to avoid Rh incompatibility. And some diseases are more common in people of particular group. Example, duodenal ulcers, that O group, gastric carcinoma, pernicious anemia in A group, in urinary tract infections in females with B and AB blood group. And about the minor blood groups includes other blood group systems like MNS, KIT, Lewy, Duffy, Lutheran, etc. And individuals who do not express Duffy antigen on RBC surface are not susceptible for malarial infection with Plasmodium virus. So that is all about minor blood group. So that with that, actually the blood group from my part is over. So you'll be having a session, integrated session on blood grouping tomorrow. That will be taken by Dr. Magdalene from Transfusion Medicine. Is that clear? Tomorrow from 8 to 9.
Okay, ma'am.
Okay, so read all these things very well because she may be asking you questions. So read all these topics very well and come prepared. Clear?
Okay, ma'am.
Okay. So if you have got any doubts, you can message me. Thank you all. Thank you.
Thank you, ma'am.
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 Okay, thank you. Bye.
Thank you.
ഇനി വേറെ സെഷൻ ഉണ്ടോ?
നാളെ ഉണ്ട്, നാളെ.
ഇനി ഞങ്ങൾക്ക് വേറെ എന്തെങ്കിലും ക്ലാസ് ഉണ്ട്? ഇപ്പൊ മഫിലാ ഇപ്പൊ കിടന്നിരുന്നത്. ഷെഡ്യൂളിൽ ഇപ്പൊ മഫിലായിരുന്നു. ഷെഡ്യൂളിൽ മഫിലായിരുന്നു. അതാ
മസിൽ അല്ല, അത് എക്സ്ചേഞ്ച് ആയതാ, കുഴപ്പമില്ല. അത് അടുത്ത ബുധനാഴ്ച ഉണ്ടാവുള്ളൂ.
ഓക്കെ, താങ്ക് യു.
Thank you. Thank you, ma'am.
Thank you, ma'am. Thank you, ma'am.
Thank you, ma'am.
നമുക്ക് ഇഷ്യൂസ് വേണ്ടല്ലോ. അവർ ഞാൻ പോയി കണ്ടിട്ട് വരുമോ? അവർ വന്നതല്ലേ, ഇറങ്ങാൻ താല്പര്യമുണ്ടെങ്കിൽ ഇറങ്ങും, ഇല്ലെങ്കിൽ പ്രശ്നം ഉണ്ടാക്കാനാണെങ്കിൽ ഞാൻ പറഞ്ഞു പ്രശ്നം ഉണ്ടാക്കാനില്ല. വേറെ എങ്ങും തുറന്നിട്ടില്ല എന്ന് പറയുന്നു. നാളെ രാവിലെ ഒന്ന് നോക്കാൻ ഇതുപോലെ